Welcome to part 2 of 3, looking at chapter 12, gene expression at the molecular level. So the first part of this chapter we kind of summarized what we're going to be looking at, so how our DNA goes through transcription, translation to make polypeptides, which are then used to create all the proteins that give us our traits and characteristics. So we ended up off on the first part looking at this molecular or central dogma of molecular biology in prokaryotic and eukaryotic cells. And again, here I'm kind of summarizing the similarities and differences between prokaryotic and eukaryotic cells at the central dogma. So again, prokaryotic cells, they can do transcription, translation in the cytoplasm. Eukaryotic cells, it's a little more complicated, which is kind of a theme about eukaryotic cells. So transcription, and then an extra step, RNA processing, occurs in the nucleus, and then that RNA, once it's processed, will exit into the cytosol, and that's where we have our translation part. And we're going to be focusing on the transcription part in this section. So just a reminder, transcription, this is where we take our DNA, we're going to produce an RNA copy, or the transcript of our gene. And structural genes produce a type of RNA called messenger RNA, or mRNA. And again, if you remember in a previous chapter in this course, there's actually 3 different types of RNA. We have messenger RNA, or mRNA, we have ribosomal RNA, or rRNA, and transfer RNA, or tRNA. This section, we're just going to see the mRNA. When we get to part 3 of this chapter, we'll see the other 2 types of RNA show up, and we'll talk about their significance in the central dogma. And then translation is where we're going to take that messenger RNA, and we're going to take it, bring it to a ribosome, and we're going to use it to synthesize a specific polypeptide on that ribosome. And again, we're really going to focus on the transcription part in this section. So transcription, we have on our DNA, we have sections called genes. So these genes are organized units of our DNA. That gene is going to be transcribed into an RNA molecule, and then that RNA molecule is going to be translated into a polypeptide. The other genes can code for RNA itself, and we're going to have transfer RNA, or that tRNA that I mentioned, and it translates, or it helps in this translation process from mRNA into amino acids. And then the other type, the ribosomal RNA, or the rRNA, makes up part of our ribosome. And again, we'll get into the tRNA and the rRNA in the next section of this chapter. So we're going to look at a gene up here. So up at the top of this image, we have a DNA strand in the pink. If we look at a gene, genes have, at the beginning of the gene, you'll have a regulatory sequence, and then you'll also have a promoter. The regulatory sequence in the green, that is where the gene can be turned on or off. So you can actually stop transcription, or you can start transcription at that regulatory sequence. The promoter, this is the region where we're going to start transcription. And then next to the promoter, we have our actual gene, which is the transcribed region that we're going to use to make our mRNA. And then at the end of our gene, we have the black terminator on the right-hand side. And that's going to signal where this transcription process should stop. Okay, so we have a regulatory sequence, we have to control the transcription, we have a start, and we also have an end on our gene. So in this transcription process, we're going to take our DNA and transcribe it into an mRNA. It occurs in 3 stages. So we're going to have initiation, elongation, and then termination. So in initiation, this is our recognition step. In bacteria cells or prokaryotic cells, we have a sigma factor that allows an enzyme called RNA polymerase to recognize the promoter region. So remember, we had that promoter region, that start of our gene. This stage, this initiation stage, is complete when your DNA strand is separated near the promoter to form an open complex. So it's kind of similar to DNA replication. We have to separate our two DNA strands and form this open complex, and then we'll have an RNA polymerase enzyme sitting at that open complex near the promoter region. RNA polymerase, hopefully you can kind of figure out what it does based on its name, but if you remember in DNA replication, in the previous chapter we had DNA polymerase. DNA polymerase creates DNA, so RNA polymerase, this is the enzyme that's going to create RNA. Okay, so enzymes, it's really nice because you can pretty much tell what they do based on their name. So use that to your advantage and remember that little bit of advice. So initiation, this is what initiation is actually going to look like. So we have our DNA strand in the double helix. Our promoter is represented as the blue section on our DNA strand. So our promoter, we have a sigma factor that finds the promoter. That sigma factor then allows our RNA polymerase to attach on to the DNA. The RNA polymerase is going to form the open complex that's going to separate our two DNA strands from each other. Once everything is set up, then we're going to have the second stage, the elongation stage. This is where your RNA polymerase is going to synthesize our RNA transcript based on our DNA. RNA polymerase, just like DNA polymerase, can only go in one direction, so it's going to synthesize this RNA molecule going 5 to 3. So one of your DNA strands is called the template strand that we're going to read to create our RNA molecule. The other strand is the non-coding strand that we can't use because it's going the other direction. Remember, DNA is anti-parallel, so one is going 3 to 5, the other is going 5 to 3. And then again, remember, because we're making RNA, you need to substitute uracil in instead of putting in a thymine, because RNA has uracil as one of its 4 bases. So on the left-hand side, we had our initiation. Now we're going to be looking at the elongation part in the middle. So what happens is your sigma factor breaks off. You can see it exiting out at the top middle. Our RNA polymerase starts to travel towards the right, and as it travels towards the right, it uses one of those DNA strands as the template strand. And then using that template strand, you use those complementary base pairing rules, but remember to put uracil in instead of thymine. So if you look at the little zoomed in white box on the top right hand side, our DNA is the red on the top, our new RNA is on the bottom in the yellow. So if our DNA has an A, on the RNA we'll put a U. If DNA has a T, RNA we put an A. DNA is C, RNA we put G. DNA G, RNA we put a C. So again, we still have those complementary base pairing rules or Chargaff rules, but we're making an RNA molecule, so really remember that part of it. This RNA is created going 5 to 3, and RNA polymerase just keeps on traveling down until we get to the third stage of this process. So our third stage is the termination stage. So our RNA polymerase is going to reach that termination sequence, which remember was that black section on our gene. And once we get to the terminator, it's going to cause the RNA polymerase and the newly made RNA transcript to disassociate or basically break away from our DNA molecule. So this is shown at the very right-hand side of this image up here. So our RNA polymerase, that purple enzyme, reaches the terminator sequence, which is shown in the black on our DNA strand. Once you get there, the RNA polymerase breaks off and our mRNA transcript also breaks off. And that's in the yellow molecule over there. So this is our first part, our transcription part, where we took our DNA information and we just transcribed it or made a copy into an mRNA molecule. So the direction of transcription, it really depends on how the gene is set up. And some genes go one direction, other genes are facing a different direction. So if you look at this piece of DNA in the pink, gene A over on the left-hand side are promoters on the left, and then we have our gene, and then we have the terminator for that. And then we have a little purple sequence, which is just DNA in between our genes. Gene B is set up the same, so our promoter is also on the left-hand side. We have our gene that we're going to transcribe, and then we have our terminator on the right side of the gene. So gene A and gene B, if we look at the DNA, the template strand is going to be the bottom strand on these two genes. Because they're going 3 to 5, that allows us to create our RNA going 5 to 3. And you can see that in the image up here. If we look over gene C over on the right-hand side, it's set up going the different direction. So our promoter is actually on the right. We have our gene C, and then the terminator is on the left. Okay, so hopefully you can see that because this gene is going a different direction than gene A or B, our template strand is now going to be the top strand. So our template strand is going 3 to 5 because we want to make our RNA going 5 to 3. Remember, it's always anti-parallel, they're always going in opposite directions. So really keep that in mind when you're taking your DNA strand. Make sure that you're going the right direction when you do your transcription. And then also remember to put uracil in instead of thymine. So that's really another big mistake I see a lot when you take your DNA strand in the homework and you'll take it through the transcription process. So eukaryotic transcription, basically it's identical to what we just looked at in prokaryotic cells, but eukaryotic cells, because again they are a little more complex, we have different types of RNA polymerase. So RNA polymerase 2, this is the one that's going to transcribe your mRNA, which is the process we just looked at. Two other polymerases, DNA polymerase 1 and 3, they're going to transcribe the genes that code for rRNA and tRNA. So each of our 3 different types of RNA, they each have their own RNA polymerase that's going to do the transcription process.